throughout the session.
Okay, great. All right, thank you. So hopefully you can hear me all right. We have a ton to go through tonight. I'm going to be updating you as best I can on all of the new research on autism. I have over 70 slides that I have to get through in 80 minutes. The reason I'm doing that is because I know how important your time is. All of you are out there. This is a precious time of the day. Most of you are clinicians or parents, and you've been very, very busy all day, and you want to get back to your family. So we have worked very hard to put the most content possible into an hour and a half. So I know I'm going to be going quickly, but you will be able to get the PowerPoint slides from Carrie. You will get them in a PDF form, and you can also, she's recording it, you can also listen again and hear all the content again if it seems like it's going a little too quickly for you. My theory is I'd rather have too much information for all of you than too little, and I also want to make sure that that we honor your time because we know how valuable it is. So with that, I'm going to get started. First, just as a disclosure, you all know, I think, that I'm paid as a part-time employee of Scientific Learning Corporation, the producer of the Fast Forward programs, and I will be talking about Fast Forward programs, but I'll be talking about other programs as well, and I have no financial or non-financial interest or related personal interest or bias in any of the other organizations that I'm going to be talking about or any of the products that I'm going to be describing or reviewing. There are several references for you. Most of the references you will see are 2010 to 2012 because the area of autism spectrum disorders is just expanding so rapidly. I have a separate page of references on computerized cognitive interventions, and you can get those when you get the PDF and look through those more thoroughly. I also want you to know of two good summary resources on autism. One is a Nature special issue that was published in 2011 on the autism enigma, but even that is 2 years outdated. There's a brand new special issue of the journal Neuron. The journal Neuron usually costs several, $35, $40 just to get one issue of it. It's several $100s if you subscribe. And this particular issue of the journal Neuron is the special 25th anniversary issue. It was published October 30th, just last week, online, and you have free access to that through November 2013. And it's like a textbook on all of the newest neuroscience, not just autism, but neuroscience in every single area that you would be interested in, and it's all free through November 30th. So definitely what you see here is a link that will take you to it, and I also want to recommend the article by McCarroll, Progress in Genetics and Polygenetic Brain Disorders, and then there's another excellent article in it that is also free, and I will be reviewing that on neuroscience technology and how we drive change in the brain in neuroscience. So those are two excellent articles, brand new, hot off the press. So probably most of you saw this in local papers. The papers came out with the news today. It was also on, I was watching, I think it was ABC News last night, and they highlighted this research by Jones and Klin on how we can now identify autism spectrum disorder in children as young as 2 months of age simply by looking at their eye contact, or actually their ability to scan a face. And the big question we have whenever we get new research like this is, why does this happen? Why is it that at birth you can't see any difference between a child who's going to present with autism and a child who isn't? But the research showed that what they did is they had little infants, and you can actually see it from the slide here. You can see the infant over here on the left in their little car seat, and what the infant was looking at from a very young age were pictures of faces of people. And what you can see down below is the children with with autism spectrum disorders, ASD, had eyes that wandered all over the face and did not focus on their eye gaze did not focus on the eyes of the speaker, whereas typically developing children focus directly on the eyes of the speaker. And this difference became more pronounced from 2 months to 6 months of age, so that the child's interest in faces and eyes decreased from 2 months to 6 months of age. And in addition to that, the more severe the variability or the variance in the eye gaze, the more severe the autism the child presented when they were 3 years of age. Now, this study will have to be obviously replicated. You might know Ami Klin well because he has been studying eye gaze and autism for a long time, and he is the individual who identified that children on the autism spectrum are not interested in action. Older children are not interested in action sequences like patty cake, and their eye gaze is different in terms of paying attention just to action. But so the question is, and what I'm going to try to answer for you tonight is, why does this happen? Why would we see a child who has what seems to be a very normal infancy for the first 2 months, they're developing normal, and all of a sudden, normally, and all of a sudden by 2 months of age, the child is starting to show variability that we can actually pick up objectively? Why does it happen, and what does it mean? And for that, we're going to go to neuroscience, because what we have in neuroscience now is a way to explain this to you. Neuroscience is always interested in why. Why is this happening? What is going on in the brain that could explain what's happening? And that's what I'm going to go through with you, and then we're going to talk about what you can do. So, as you all know, autism spectrum disorders, just so we're all on the same page, are developmental disorders characterized by impairments in 3 areas. The child has difficulty with social interaction. The child has varying degrees of verbal and nonverbal communication deficits, and they have restricted, repetitive, and stereotyped patterns of behaviors and/or interests. Autism spectrum disorders are neurodevelopmental. We know that. It is the brain of the child with an autism spectrum disorder that is different, and our interest in neuroscience is to understand why it's different. You probably all know that autism spectrum disorders, the incidence of autism spectrum disorders is now 1 in every 88 children. That has decreased from 2009 when it was 1 in every 110 children. So, or increased, sorry. So the incidence is increasing dramatically. And it's greater than 1 in 70 in every male, in males, so that autism spectrum is much more common in males and females. And incidentally, that research you just saw that was just published yesterday online in the journal Nature they had 11 boys that they followed from birth all the way through to 3 years of age. 11 of the 100, I think over 100, almost 150 children they followed, I believe, or 110 to 140. I can't remember the exact number because I just read it quickly today. But of those, 11 who were high risk, that is, who came from families where there was a sibling on the autism spectrum, 11 did end up by 3 years of age presenting with autism spectrum disorder, whereas only 2 of the girls in the group did. So we know that it's increasing, and we know it's more common in males. In the past 2 years, what we have learned is that we've known for a while that it is a genetic risk. We call it a polygenetic disorder, and I'm going to explain that to you. But it is caused by genetic mutations that occur oftentimes in the child, him or herself, not in the parents necessarily. But we'll talk about that. and I'll explain that to you. In the last 2 years, we've clarified what those genetic changes do, and essentially what they do is they have effects on brain structure, which doesn't surprise you at all, and especially white matter tracts. So I'm going to explain that to you. And then in the last two years, what we've learned is that the value of targeted therapeutic interventions, and by that I mean computerized interventions that are very well structured and designed to drive neuroplastic changes in the brain, combined with, and that's why you see the plus there, standard individualized treatment that addresses the specific needs of every child that you see, because this is a very heterogeneous disorder. The combination of those two strategically build language and social skills, as well as there's evidence of white matter tract development that I will share with you tonight. So just to get us started, I'd like to start with this particular view of the human brain to remind you, all of you who are providers of Fast Forward, I know are aware that the brain has 4 major lobes, 3 sensory lobes, the parietal lobe, the occipital lobe, and the temporal lobe. And then about 1/3 of the brain is the frontal lobe. And the frontal lobe is the part of the brain that acts on the world around us. The sensory lobes pick up information. Now, there's a very important fiber tract you're going to see. There are lots of important fiber tracts, but there are several that are important for language that run from the occipital lobe through the temporal lobe to the frontal lobe. Those fiber tracts are quite long. So if you can imagine, the analogy in the United States would be a highway system that runs all the way from Maine to the tip of San Diego. And the longer a highway system is, the more vulnerable it is. And the other thing you need to understand is it takes longer for it to develop. So just as the highways that link the East Coast to the West Coast of the United States took many, many, many years to build, and we had many small highways before we had the huge interstate, the same thing happens in the human brain, so that they are vulnerable. And that's what we're going to talk about tonight. Now, the reason that we weren't able to see these fiber tracts until very recently is we didn't have a technology that allowed us to do this. But in around the middle of the 2005, 2006 era, a new kind of MRI became available, magnetic resonance imaging, called diffusion tensor imaging. Prior to diffusion tensor imaging, we could look at an image that you see over here in number 1 and number 2, which might give us a CAT scan or an MRI of the brain so that we could see basic gray matter, basic white matter, but we really couldn't see clearly fiber tracts. With diffusion tensor imaging, what we're actually doing is we're measuring the diffusion or the motion of protons that are in water molecules. And so what that allows us to do is we can actually very specifically map out these white matter tracts, because white matter is myelinated, and myelin is loaded with fat, which every woman probably that's listening to this knows is loaded with water. So we all know the fastest way to lose weight, if you're a woman, is to not drink anything for about 2 days, and then your fat cells, which are loaded with water, will deplete their water supply and you will lose weight. The problem is, of course, once you drink water again, you'll gain the weight back and you haven't lost fat really. But so fat is loaded with water. And so by being able to visualize the fatty myelin, we're able to really get a good picture of myelinated fibers. And once we could do that, we could start looking at children on the autism spectrum and delineating the differences in myelination and in their white matter tract development. And that's what changed this whole discussion. Now, when you're looking at white matter tracts, it's called fractional anisotropy. You can see that word here. And we're going to be talking about that because when we talk about the white matter tract development, you're going to see it varies in children at different ages that are on the autism spectrum. So we're going to talk about that. And that explains why eye gaze, for example, might be normal for a few months and start to change after the child is developing. And I'll get into that in a little more detail in a minute. So one of the things we were able to observe around 2006, for example, is how this fractional anisotropy, here you see the FA down here, that's what it's referred to as, how that enables us to actually visualize these fiber tracts and their maturation in children from 12 months to 48 months. And what you can see in these drawings is how the fiber tracts get thicker and they get more consistent. So there don't seem to be little gaps in them. And you see as much development over here, for example, in the 48-month-old where you see my little red dot, you see as much over here that is short fiber tracts. Those are just the little fiber tracts that are linking regions that are closer together versus the very long tracts. The long tracts are referred to as longitudinal tracts, and that's important. Now, these are some of the longitudinal tracts that you're seeing here, and this was research published by LaBelle in 2008. And interestingly, I was just speaking in Edmonton, Canada, a couple of weeks ago, and I presented this research of LaBelle's, and it turns out that LaBelle actually did this research in Edmonton, which I didn't realize, and I understand now LaBelle is now at UCLA. But what you can see in this particular diagram is that all of these longitudinal fiber tracts are maturing at different rates. And what's important for us understanding autism spectrum disorders is that these different rates of maturation, and this is a very little picture, but if you look at the bottom of each of those graphs, you'll see that the ages are from 5 to 10 to 15 to 20, 25, 30 years of age. So we have some fiber tracts, like our cortical spinal fiber tract, this light blue one up here, or the uncinate fasciculus, which is this yellow one down here, that are actually maturing into our 30s and perhaps even longer. The uncinate fasciculus is very important for prefrontal lobe function, especially things like editing and outcome monitoring. So we have some skills that continue to mature into our 30s and 40s, and that's because the fiber tracts are maturing at that rate. Now, the first person who really identified that autism spectrum was a disorder of fiber tract development, a lot of people speculated that it was, because you could see on fMRI, for example, in the 2000s, you could see that areas of the brain in children on the spectrum were not linked to each other the way they were in typically developing children. But WAS had an article in 2011 where they actually used DTI, and they did show that, that there was this structural underconnectivity in mature subjects with autism spectrum disorders. And the question then is, and neuroscience and neuroscientists always ask why. So why? What is causing these fiber tracts not to mature? And so a lot of research was looking at causes that could contribute to fiber tracts not maturing. And parents looked at things like diet, and parents looked at things like immunizations, which we've since learned are not issues in this fiber tract development. Not that diet isn't important. Diet is important for your brain development, for your body development, but it doesn't seem to be the reason why these fiber tracts aren't maturing. So the causes turned out to be genetic, and what we specifically call polygenetic. And I want to explain genetics to you. It's a little complicated. It's taken me a long time to understand genetics. I had to take almost a few courses myself in it to start to understand it, because it's not the way I was taught genetics, which is Mendelian genetics, which is if I have blue eyes and my husband has brown eyes, I'll have 3 brown-eyed children and maybe 1 blue-eyed child because it's a recessive gene. That was the genetics I learned. But new genetics really is much more complicated, and that's what I want you to understand. So autism spectrum disorder turns out that there are these 4 issues. One is that it does look like some families do have a autism, a susceptibility to having a child on the autism spectrum because they have susceptibility genes. And I'll talk about that, but that doesn't seem to be one of the primary causes of autism. Primary causes of autism seem to be copy number variations, which are a kind of mutation where sections of the genes are copied incorrectly. I'll explain that to you. And or point mutations. Point mutations are mutations in a gene where one of the little letters, the sequence of the gene, gets misplaced. And you'll see that, and I'll show that to you. Now, there are genetic syndromes like Fragile X and Rett syndrome that are unigenetic or monogenetic, meaning that those are caused by single genes, and they cause the syndrome of which autism can be a component. But we're going to be looking at the polygenetic varieties. So, as I said, this is the hand-wringing that's kind of typical of little girls who have Rett syndrome, and we know that those genetic syndromes are simpler in the standpoint that there are specific genes that are associated with them. But we're talking about the vast majority of autism spectrum disorders are caused by genetic mutations that are very complicated. So I just want to explain this to you briefly because it's important when you understand treatment and you understand where treatment is going to be effective. So the genes in your body, you know, are constantly replicating. And you know that when a sperm and an ovum unite, that it creates a combination of the dad's genes and the mom's genes. And then those cells start dividing. And every time cells divide, the genes have to replicate themselves. And whenever they replicate themselves, there's a chance for an error. And so when you get a copy of a gene, it's called an allele. And the alleles are where the errors occur in the copies, just like when you are using your copy machine and you run out of ink, you end up with a copy that doesn't have all the information on it. Well, the same thing can happen when genes replicate themselves. So there are many potential sources of errors in replication, and there are many disorders and diseases that occur when this replication goes awry. So cancer is a good example in all of us as adults, for example, and then some children, unfortunately, where cells replicate themselves, make mistakes. Those mistakes go unchecked, and the cells start dividing in a rapacious way and create tumors or other kinds of abnormal cell structures. So cell errors in replication, these genetic errors can occur at any time in life, any place, whenever cells are dividing. Now, if you inherit a mutation, so I could have mutations in my own genome, and sometimes they could be caused by environmental influences or other issues, stress perhaps, and then I could pass some of those changes on, those mutations. So those would be heritable. But you can also have de novo mutations, mutations that occur in the child that didn't occur in the parents. And it looks like a lot of these de novo or new mutations are the factor in autism spectrum disorder. So just so you can see what copy number variations look like, this is a gene duplication that created a copy. You can see it over here on the right. And in this copy, one segment was duplicated twice, and it shouldn't have been. So that's one source. A copy number variation, or CNVs, are one source of mutation. Now, point mutations, as I said, are where a single letter within the genome sequence gets replicated incorrectly. And there are basically two kinds of point mutations. One is called a missense mutation, which I'll show you in a minute. And diseases like sickle cell disease and amyotrophic lateral sclerosis are caused by missense mutations. And that is a mutation in which the little nucleotide change results in a code that creates a different amino acid. Nonsense mutations are point mutations that result in the code stopping itself. And I just want to show you each of these. So what you see here is the 3 kinds of mutations. And what you're looking at here now, over on the right-hand side, is what we call the codon, the RNA codon. So that is a series of letters. And in RNA, the letters are A, U, G, and C. Now, in DNA, the letters are T, C, A, and let's see, what am I missing? T, C, A, and G. So what can happen is you have these little letters, these little codons, or this little sequence, and it produces an amino acid. So what you're looking at over on the left is I have the DNA TTC, or the RNA AAG, and that produces lysine. Lysine is just an amino acid, and I don't know if many of you ever supplement your diet with amino acids, but amino acids are what make proteins work in your body and make your body work. So amino acids are very important. Now, you can have 3 kinds of point mutations. You can have a silent mutation where there is a change, so TTC changes to TTT, but it doesn't really do anything. You still end up with lysine. But in the nonsense and missense mutations, what happens is a different, in missense, a different amino acid is produced. So if TTC becomes TCC, then you have arginine instead of lysine. And if TTC becomes TGC, then you end up with tyrosine, for example. So that's huge in the body to end up with a different amino acid in a gene than you intended, because then that means that that gene is going to work differently. It's going to work differently in your body, and it's going to do different things. And so a lot of us have mutations. We all have mutations going on all the time, and there are a lot of very common mutations, as you can see down here, that occur in us, and they don't seem to do any harm. We have lots of checks and balances in our system. But when you start having rare mutations over here in the dark blue, and you have a lot of them, that's when autism spectrum can occur. So we know from a wonderful article published by Sanders in the journal Nature just this year, just not too long ago, and it's in your bibliography, that it's these de novo mutations that have been now revealed by this procedure called exome sequencing that are associated with autism spectrum. So what's important about this is that children on the autism spectrum have more mutations. So you can have lots of mutations. You can have 3 mutations, and you can see in a number of cases, 3,000, no one was on the autism spectrum. But when you start getting these multiple mutations in children, you increase the risk, especially if they are rare mutations. And what Sanders then concluded is that it clarifies that ASD is a genetic disorder, and it doesn't necessarily mean that the parents produced or had the gene, that the child has a gene that the parents had. It just means that something went wrong with the genes, often just in the case of the infant themselves. It's very heterogeneous because you have so many mutations occurring in children on the autism spectrum. So if you have dozens and dozens of mutations that occurred in genes in one child and then different ones in another child, even though they're both autistic, they're going to be very different. So we have heterogeneity in terms of the genetics, and we have heterogeneity in terms of the children themselves. And oftentimes I'll say, in my whole career, I've never seen 2 children on the autism spectrum who are exactly the same. And I think if we look around the entire world, we would find that there aren't probably more than one or two in the world at one time that would be exactly or would have exactly the same symptoms. And then when you think about the brain changing over time, that complicates it even more. So it is these disruptive de novo single nucleotide variants. So those are those little point mutations that cluster robustly and identify children on the autism spectrum. It's important when we finish up this section on genetics, though, to understand that gene mutations are not a bad thing. They're the way that our body adapts to our environment. So just as a quick example, there are people who are lactose intolerant, and every time they drink milk, they get indigestion from it. But there was a whole group of people who raised cows in Europe in certain areas, and milk became the primary source of protein. And so their cells over time were able to develop a mutation so they could tolerate cow's milk. So sometimes mutations are very, very helpful. They allow us to adapt to changes in our environment and survive in the environment. But it's when you have the rare mutations that we're talking about that confer the risk of autism spectrum disorder. Now, second very important factor, and it gets us back to neuroscience, there are two very adaptive parts of the human body. One is your genome. Your genome changes based on the environment, and that's what these mutations are. But your brain is also an adaptive organ in the human body. So the brain is changing rapidly as well based on the environment. And so you can start to imagine that if you have a plastic brain, a brain that's very changeable, and you change a lot of genes, that one of the major things those genes are going to affect is the human brain. And it turns out that's what is causative in autism spectrum disorders. So to summarize, autism spectrum disorders is very complex and very heterogeneous because we have all of these genes that start to enter in any one child, or certainly many genetic mutations, and those are going to affect the brain in unique ways. And they seem to have primarily a major effect on developing fiber tracts. So the primary part of the brain that's developing after the first trimester, and you probably know this, is the fiber tracts. In the first trimester, what's happening is your brain is being laid down. First trimester of pregnancy, all the neurons are being formed, and you'll get most of the neurons you're ever going to have in your whole life. Then the second trimester, the neurons migrate to form the spinal cord and the peripheral nerves and the cerebrum. And then the third trimester on through 30 years of age, neurons are connecting with each other, and these fiber tracts are being laid down. Now, there's a wonderful study published by Wolf in 2002 using DTI, updating the research that I presented from WAS, showing the trajectories that we see with fractional anisotropy for high-risk autism spectrum high-risk groups. And you can see one of the fiber tracts that seems to be affected more frequently in autism spectrum disorders and in autism in children that have high risk for autism is the corpus callosum. And there are three different parts of the corpus callosum that are affected. The genu is this anterior part of the corpus callosum that links the right and left hemisphere together in the prefrontal cortex primarily. The body of the corpus callosum, very important for coordination of your body. And then the splenium, very important for vision and coordination of vision from right and left hemisphere. But in addition to the corpus callosum, we have many other long fiber tracts. And you can see them here. The interior or the inferior longitudinal fasciculus here that links the occipital lobe runs inferiorly in the brain to the frontal lobe. The uncinate we talked about, very important for prefrontal lobe function. And the fornix are fiber tracts that have been identified as being affected in autism spectrum. Now, here's the point, and this now gets us back to this new research on eye tracking in children 2 to 6 months of age. you see a distinct and pervasive aberrant course of white matter fiber tract development in children who are at high risk for autism spectrum disorder and went on to develop autism spectrum disorder. And those children who are at high risk are children who have siblings with autism spectrum disorders. Now, here's the key. Most of the fiber tracts of the high-risk infants were characterized by higher fractional anisotropy. In other words, the fiber tracts were thicker at 6 months, followed by blunted developmental trajectories such that the fiber tracts, the fractional anisotropy, that is the thickness of those fiber tracts, was lower by 24 months. Now, here's what this means, and I want to make this really clear to you, is that what happens with autism spectrum disorders is the brain starts out developing normally, and then those fiber tracts start to have abnormal development. The child isn't born different. So parents are right when they say my child was developing normally, and then gradually I started to see these behaviors that were different, and that is true, and it's because of the fiber tract development. And the core, and this is Wolf, that same fiber tract study, the core behavioral manifestations in autism spectrum disorders are due to atypical patterns of connectivity that are caused by genetic mutations. So it is white matter tracts that don't develop normally caused by genetic mutations. Now, they differ across systems and time, so every child's going to be a little different. Some children, that the problems are going to be obvious a little earlier, some later, some in some fiber tracts, some in others. And there's not a specific brain region or behavioral domain that's affected. Okay, now, what causes? Remember, scientists in general always ask, what causes? So what causes the genetic mutations? Remember, we said our body is designed to mutate. The genes are, my genome is designed to change based on the environment so that I can adapt to changes in the environment that occur. So genetic mutations then are likely to occur in response to changes in the environment, and that's number one. So new chemicals in the environment that we haven't had before, petrochemicals that are in the environment, chemicals associated with pesticides that are new pesticides that we haven't had in the past, chemicals associated with production of synthetic substances, all of those chemicals that are in our environment that are changing because technology changes, those introduce the possibility of affecting our genome. Secondly, age of parents is an issue, and there's a reason for this. A mother is born with a mother has all the eggs when she's maturing that she's ever going to have. So if a mother has ages, what happens is those eggs age and they get older. And as an egg ages in the mom, in her ovaries from, let's say, 18 years of age to 40 years of age before she finally just doesn't have any, she doesn't ovulate anymore, those genes inside of those eggs can deteriorate. And one of the things we've known for years that can be caused by an aging mother is Down syndrome. So Down syndrome is a genetic abnormality or genetic event that occurs in the mother's egg, and we can test for that with amniocentesis and determine if a baby has Down syndrome. But it turns out that the father is producing sperm for his whole procreation lifetime. And so unlike the mother whose eggs get old, the father's eggs per se, or the father's sperm don't get old, but he gets less good at producing them as he ages. And so remember that genes are always replicating themselves. So when a dad is producing his sperm, those sperm are replicating the father's genes and the genome to pass on, and errors can occur. So the fact that parents are aging seems to be another factor that's contributing to more of these genetic mutations that are occurring in infants. But I want to remind you again, I don't want people to become panicked. We all have genetic variants, and many of them are not problematic. So in autism spectrum disorder, it seems to occur when too many of these variants occur. So it's a issue of number primarily. Now, we also know that there may be other issues in autism that are driving either genetic changes or driving abnormalities in the way the body or the brain develops, especially. And in addition to parental age, in addition to chemicals in the environment, we have research that over the last 20 years that there may be immune factors that play a role in the etiology of some forms of autism. Now, what do I mean by that? Well, if mom has a cold or a virus of some kind, normally the baby is protected and nothing happens. But mom, when she has a virus, produces antibodies, and those antibodies can end up in the amniotic fluid and affect the way the baby develops. So Judy Vandewater and Paul Ashwood, who are colleagues at the Mind Institute, have evidence that immune factors may cause inflammation and may cause inflammation in the developing fetus or in the child in utero, and that could contribute to autism spectrum disorders. And so I want to draw your attention to that research. It was published just a few years ago, or just a few, months ago, in the journal Science, this inflammatory hypothesis that Vandewater and colleagues have developed, that these antibodies in a mother's bloodstream travel through the placenta, cross the fetal blood-brain barrier, interfering with specific proteins that could lead through multiple steps to problems with brain development and maturation. So that is another source of possibility explaining at least some children on the autism spectrum. And the Mind Institute has developed an autism test to look for these antibodies because if a mother had those antibodies and had one child on the spectrum, it might be the reason she would have a second child on the spectrum. So parents soon will be able to be tested for these antibodies. As you can see from this slide, there is some concern over this test because they don't want to scare parents unnecessarily. But that test will probably be available. Again, it's important also to understand that that would only explain a small proportion of children on the spectrum, likely, so that just because a mother, took the test and it was all right, doesn't necessarily mean that there would be a child that wouldn't have issues. So to summarize, the current view is that autism is polygenetic. There are multiple genes that affect the development of long fiber tracts that support language in the left hemisphere and social skills in the right hemisphere. So let's look at those fiber tracts now. Now, I'm going to look at the two that are most important for speech and language that were identified by Turkin and Dronkers in research they did with adults. So they did research with adults who had had strokes, and what you can see is that, that in adults who have strokes, where the region of interest is here in the inferior frontal gyrus. Most speech pathologists know this as Broca's area. There's a long fiber tract, the inferior longitudinal fasciculus, the inferior occipital frontal fasciculus, IOFF. Here you can see it. It's purple. It's a primary fasciculus, one of its own, very long, runs all the way from the back of the occipital lobe all the way forward to the front of the frontal lobe. Very important for speech, fluency, and grammar. So if that didn't develop, whether you had a child on the spectrum or whether you had a child with other issues, if that fiber tract doesn't mature, then we're likely to have a child with speech, fluency, and or grammar problems. Now, Turkin and Dronkers identified white matter tracts underlying auditory speech perception, and this is all very important for all of us who use programs like Fast Forward, for example, because speech perception has a lot of fiber tracts associated with it. They also are quite long. They also are quite complex and overlap and run inside of each other and outside of each other. But the important part of these fiber tracts is that they are associated with auditory speech perception. So obviously, just if we don't do anything else, if we have a child who has speech and language problems, the chances are that we are going to have an auditory perceptual component of that, and that we're going to want to have to address that therapeutically. And we'll get on to that more later. Now, there's also the social network of the brain. Primarily, the part of the brain that handles social skills is right hemisphere. The social network, though, also requires long fiber tracts. And there's this wonderful article that you can get for free for the next month from the journal Neuron, Toward a Neural Basis for Social Behavior. The link is there, that talks about the social networks. It's an article by Stanley and Adolph, just was published about 5 or 6 days ago, October 30th. And what you see is that the right hemisphere is very important for social skills, and that we have several long networks. We have a mentalizing network that's very important for theory of mind. That's taking perspective of others. That's this blue one here. You can see it. The amygdala network, which is down, you see at the base of the brain, very important for handling emotions and controlling our emotions, involves frontal lobe control, self-control, and emotional regulation. And then the empathy network, this dark blue network, very important, involves the prefrontal cortex, very important for the ability to care about others. And we also have the mirror neuron network as well. So this green network that you see here. So the right hemisphere has long networks also that are long pathways that are very important for social skills. So now here's the good news. The good news is that because everyone is agreed on one of the features of ASD is that it affects white matter development. White matter development is set into motion, and in autism spectrum disorders, it might be arrested by the genes. Okay, now we can't do much about the genes, at least not yet. But the good news is white matter is highly under the influence of environmental stimulation, so that white matter tracts, unlike neurons, that you're born with the neurons that you're going to have most of your life, and you just have to kind of live with that. The white matter tracts are neuroplastic. They are amenable to training. They are amenable to environmental stimulation, which means that ASD is a set of disorders that should be highly affected by intervention. And one of the ways we can intervene and one way we can drive fiber tract development is through computerized interventions. And that's what's so exciting about this, because computerized interventions are ways that have been designed through years of neuroscience to drive fiber tract maturation and development. Even in a damaged brain, like in a phasic brain, as you know, we have posit science, or we have areas of research that are coming out, and I'll explain some others of these that have been designed to reroute fiber tracts even in a damaged brain. And in a child on the autism spectrum, we're not talking about a brain that's damaged, we're talking about a brain that isn't developing normally. So the computerized interventions offer tremendous promise. So what therapies are effective in autism spectrum and why? Well, we do know, I mean, the one personally interactive kind of therapy we know that we have good research on, random control trials, is applied behavioral analysis, which is a very organized stimulus response kind of therapy. Most of you who work with children on the spectrum know ABA approaches like LOVOS or the TEACH program if you're in schools, and those are programs we know are effective for changing behavior. Now, what we're interested, though, is going more than changing behavior. Yes, we want to change the autistic child's behavior, but we also want to improve their sensory and perceptual systems, which I'll explain to you in a minute. So we know, and I have quite a bit of research to show you, that computerized programs can accomplish changing perceptual systems in a very systematic way. And so they have a tremendous power over our perceptual system, which is excellent for children on the spectrum. For all children on the spectrum, driving longitudinal fiber tract development is essential to build the left hemisphere language and right hemisphere social networks. So we know computerized programs are designed to drive neuroplastic changes. That's great because they're designed that way. They're designed by neuroscientists. They're tested for that purpose, often in animals and then again in humans. And then for children on the autism spectrum, enhancing language skills and social skills as behaviors is essential. And although some people use ABA techniques to do that, looks like right now a combination of computerized interventions and clinical approaches hold the greatest promise. So I want to go through these. Now, let's start with why should we focus on perceptual problems? What do we know from neuroscience about perceptual problems and what difference does it make? Well, it turns out, and there's another couple wonderful papers on this for you, but irrelevant stimuli capture attentional resources. So if I am sitting in a room and I'm trying to learn, you guys are all listening to me on the computer and you're trying to pay attention to me, but it's late at night and you're probably hungry and you might have children running around your house screaming and your husband might say, what the heck are you doing in there? And all of those irrelevant stimuli going on in the environment are capturing your attention. And when they capture your attention, you can't pay attention to me. So if we have information coming into our brain that's incorrectly coded because we're paying attention to the wrong things, then what happens is that increases activity in regions of the brain that are going to take our attention in that direction. And actually, that research I showed you about that Ami Klin participated in, Ami Klin has suspected that the reason children's eye gaze varies is that they are paying attention to different aspects of the stimuli. And if they're paying attention to different aspects of the stimuli, if they're not paying attention to the eyes, they're paying attention to the mouth of the face, or they're paying attention to the ears of the face, or they're looking at objects in the background, then they aren't going to relate to human beings. And it's not because they have something wrong with them, it's that their attention has been misguided. So perceptual problems create an unstable state in the brain, which leads to inappropriate learning. And this is just some slides of the Neurobiology et al. That were published in 2012. You can get access to that through that new articles, the articles that I told you about in the journal Neuron. And what they're trying to show you here is in order for the prefrontal cortex to engage in efficient decision-making and adaptive behavior, the brain has to continuously make accurate predictions about the future, and those rely on rapidly, accurately comparing high-fidelity perceptions. So if you're just thinking about it from the standpoint of fiber tracts, perceptions here are linked to the frontal lobe through long fiber tracts. We want to exercise those fiber tracts, and we want to start them moving and developing through accurate perceptions. If lower level perceptual or attentional processes are degraded or abnormally biased, as you see in the children, in the infants who are looking at the wrong part of the face, that means that that perception has been abnormally biased to something different, then the brain has difficulty adaptively performing to more complex operations. So what we know then is perception is essential to getting the brain fiber tracts started, especially these longitudinal ones. Now, what else do we know about driving white matter development? We know, and again, this is Vinogradov, 2012, which is cited in the Neuroplasticity article in the Neuron journal that you can get for free, the special 25th anniversary journal. But these are quotes from that article. The prefrontal cortical association areas are uncommitted at birth, programmed to be shaped over a lifetime by the individual's unique perceptual, cognitive and affective experiences. Now, think of what happens if you have an infant that is perceiving the world incorrectly, and that starts around 2 months or 3 months. What will happen then is this high degree of learning-dependent brain plasticity, combined with the availability of computerized technology, allow us to deliver the right stimuli. Computers then give us the opportunity to take a child who's perceiving the world incorrectly and change that perception by magnifying what the child is supposed to be paying attention to drive learning events in the brain. Now, the Vinogradov study was published in the journal Neuropsychopharmacology because one of the interest there is schizophrenia, how we can use computerized technology to change schizophrenia. But I also want you to think of computer technology in the developing brain as well, and Vinogradov does refer to that in Driving Light Matters. And the key, and this is what's so important for all of you to understand, and I don't want to de-emphasize this at all, is that the key is balancing high-level processing with mid and low-level processes. That means you need computer programs that work on low-level processes like perception, mid-level processes like perhaps grammar and vocabulary, and higher-level processes that work on problem solving. So you want computerized approaches, if you're going to drive white matter, that work on perception plus, let's say, language, if that's the fiber track you want to work on, plus problem solving and executive function skills. So the critical active ingredients of neuroscience-informed approach to cognitive intervention are: 1, precise engineering of stimuli and tasks to improve the speed and accuracy of relevant information. Those of you who know fast forward, I want you to think of Sky Gym, okay? Highly intensive training schedules of carefully controlled and constrained learning events along with individualized adaptation of task difficulty that drives learning and preserves reward schedules. All of you know that well-designed neuroscience programs do exactly that. Many, many responses that adapt to the child as the child goes. And then finally, in order to maximize enduring plastic changes in the cortex, the learner must attend to each trial of learning event. You've got to grab their attention on a trial-by-trial basis. And you have to have a high proportion of learning trials that are rewarded immediately within 40 milliseconds of the person responding to the stimulus item. Those, of course, are the ingredients of well-designed neuroscience programs. Now, we know that the Fast Forward programs are targeted to work on these skills in the left hemisphere networks. What about the visual component of ASD? We just saw a huge research article that shows that that is an early identifier of children with autism spectrum disorders. So Hopkins, in 2011, who's at the University of Alabama at Birmingham, performed a well-controlled random control study of 5 hours, that's all, of computerized training of eye gaze detection. Now think about how ahead of the time Hopkins was, because we didn't even have this new research out yet, although we've known for a while that eye gaze is an issue, with 49 children with autism spectrum disorders. And they reported improvements in trained domains as well as improved social interactions, indicating that improvements in relatively low level, remember you want low, mid, and high level, low level and mid-level processing of facial features and emotions can show real-world generalization effects. Now that was published in 2011, November of 2011, so just 2 years ago, in the Journal of Autism Developmental Disorders. And the title of the article is Avatar Assistant, Improve Social Skills in Students with ASD Through a Computer-Based Intervention. The approach is restorative in nature. The idea is not to develop compensatory workaround strategies for impaired visual gaze, but rather to restore normal or typical gaze and to drive the impaired neural systems in the direction of normal functioning. All right, now let's go back to the left hemisphere and look at what we know about what we can do for the auditory processing component of auditory spectrum disorders for speech sounds. And if you went to Visionary Conference a couple of years ago, you heard Nicole Russo talk about a study that she conducted with Nina Kraus, who, by the way, just is doing research now on poverty and auditory processing. But Nina Kraus is a colleague of mine at Northwestern who has been very involved in trying to get all of us to understand how important auditory processing skills are in everything we do in the world almost. I mean, just like visual processing is very important to social skills, auditory processing, according to Nina Kraus's research, is important for reading, it's important for learning in school, it's important for social skills, it's important for attention, it's important for language. And Nicole Russo, who worked with Nina at Northwestern, actually did a study of children with autism spectrum disorders, which she published in 2008. Children were 7 to 13 years of age. There were 21 of them, 19 males and 2 females, and she looked at typically developing controls, 21 of those, 13 males and 8 females. All of the children had mental ability within normal limits, normal hearing, and no other issues, neurological issues besides autism spectrum disorders. And she looked at all sorts of parameters of auditory processing, and even just the ability to encode changes in fundamental frequency, which is the basic tone of your voice that goes from high to low. So a typically developing child, if I go, ah, like that, or ah, we see their brain match that response. But in autism spectrum disorders, if I go, ah, the child is hearing, ah, and if I go, ah, the child is hearing, ah, ah, ah. So the brain is not perceiving the sound that's coming in. It is not duplicating it. Russo investigated the effects of fast-forward training on children on the autism spectrum for 5 to 10 weeks with fast-forward. And she had a trained group of 5 children with autism spectrum disorders, a control group of 6. And what she found was that in evoked brainstem responses and cortical responses, there were significant changes from pre-test to post-test with a standard deviation criterion that was deemed significant. So these were 2 of the subjects, number 10 and number 16. This was their ability to track this pitch change, before, that's pre, and then pitch tracking after they went through fast forward. And you can see it's almost typical after just a few weeks of training on fast forward. So she summarized saying, training with fast forward produced measurable improvement in auditory function in some children with autism spectrum disorders. 3 of the children showed a change in the brainstem responses, 1 child in the brainstem response on yacht, and 5 children showed it in the cortical responses. And the cortical changes may precede behavioral or brainstem changes. So consistent with what we're now learning about the importance of perceptual processes, first you change the perceptual process, then the behavior comes afterwards. So what are some other promising technological approaches for social skills? I'm going to go back to the right hemisphere. There is a program called VSIP, Virtual Environment for Social Information Processing, also developed by Nicole Russo, who's now has a hyphenated name. She's at the Rush Neurobehavioral Center in Skokie, Illinois. It's for children 8 to 12 years of age with ASD. It is virtual reality, and she's in the midst right now of getting subjects. So if you live in the Chicago area or you know anyone with autism spectrum disorders who would like to participate in this research trial, you can let me know, and I would be happy to forward your information to Nicole on that. Now, going back to this driving of neuroplasticity, Vinogradov, in that 2012 paper published in the journal Neuropharmacology, that you can access actually if you get that New Neuron journal, because it's discussed in that article, and there's a link to this article, summarizes and says, these studies in children show that repetitive training, implicit learning that focuses on relatively lower level impairments like perception, can result in eye gaze, I should mention both of those, can result in clinically meaningful generalized improvements in real-world behavior and affect. There's also extremely high clinical potential of carefully constructed higher-level cognitive training based on an understanding of systems neuroscience. So that takes us now to the Fast Forward programs, and I just want to encourage all of you to continue to use Fast Forward with children on the spectrum, and to use it as soon as you can with children on the spectrum, because it is very powerful for driving these left hemisphere skills that were associated with those long fiber tracts, both for perception and for speech production. This was a study that came from the original research trial with Fast Forward that was done many years ago using the SELF test. And this was students who had ASD after Fast Forward use. We see a very significant improvement in the language skills of those children. Now, Marcy Melzer and Gigi Poglich also did a study of Fast Forward in a more global sense. And remember, the recommendation now is that you have low-level, mid-level, and high-level stimulation in computerized interventions. And that's exciting because Fast Forward language and then the reading products actually go from very low-level auditory perceptual skills, processing speed, all the way up to high-level syllogistic logic reasoning, paragraph comprehension, high-level reasoning tasks. So you have a wide range that you can use with the children on the spectrum. But this was a study done by Marcy and Gigi several years ago. They it's a retrospective study, meaning it was not designed before Fast Forward use. They contacted 34 professionals around the country, the U.S., who had used Fast Forward language with children. They ended up with 100 children on the autism spectrum. The criteria were that the children had to have worked with Fast Forward for at least 20 days. They had to have been diagnosed as on the autism spectrum by a medical professional. So they had to have a formal medical professional diagnosis of autism spectrum disorder. And they actually had to have two kinds of information from the children they were looking for: age-equivalent test scores before and after treatment, and the results of functional skills checklists that parents and teachers filled out before and after. And I'm just going to show you three of those major results. The first was changes in language after Fast Forward training. They had an 86 response rate, meaning 86 out of the 100 individuals did respond that they saw, not that they saw changes necessarily in language, but did respond to the questionnaire and had test data. 83% of those 86 children showed improvement in language. And the areas of language were language response time, following directions, understanding humor, overall comprehension, imitation skills for words and phrases, use of new vocabulary, lengths of utterance, answering yes/no questions, answering WH questions, who, what, where, why questions, improved verbal fluency. That's that long inferior occipital frontal fiber tract, which is so exciting, even though Fast Forward language doesn't work on verbal fluency at all, and overall expression. They also, with an 88% response rate, meaning 88 out of the 100 individuals, they had responses on social skills. And what they found was that 76% of the children, even though Fast Forward really focuses primarily on the left hemisphere skills of language, that they saw improvements in eye contact. Now, this was by questionnaires, addressing people by name, being able to maintain a topic, appropriate attention-getting devices, greetings and closures were more appropriate protesting or refusals as opposed to just using temper tantrums, ability to verbally negotiate, and initiation of communication. So those are remarkable social changes that were driven in 76 out of 88, 76% of 88 individuals who responded. The conclusions then from all of the research I've shared with you is that early diagnosis followed by early intervention is likely to be the most effective therapy. So I just want to take these conclusions and slow down for just a minute with you so that you can get a full sense of what we have learned today. Autism spectrum is identifiable probably within the first few months of development. Now, one of the things that they caution about this research that was done by Quinn and colleagues is that a parent can't look at eye gaze and discern the difference between a child who's autistic and a child who's not autistic. They used very sophisticated technology to track eye gaze in these children and to be able to determine the degree of variation from typical development. But certainly we do have some excellent early identifying characteristics that you can go to a psychologist if you have a young child, an infant, and the psychologist can look at some of the early signs of autism spectrum in early months, and hopefully soon there will be this eye gaze test available that if it looked like you were concerned or if there's a high risk for autism for some reason, older parents, or perhaps other children in your family on the autism spectrum, that you'll be able to perhaps diagnose this quite early. And the antibody test also holds promise. So the earlier we can diagnose children on the autism spectrum, the better, because then we can get involved right away with early intervention. Now, it's important to stress that in the early 2 or 3 years of development, computerized interventions really aren't available yet. By 3 years of age, you can use computerized interventions, but 3 to 4 years of age, depending on how severely, how high or low functioning the child is on the autism spectrum. So in the early years, what we know we have to do is a lot of interacting with the child. We need quiet environments. Children need to hear a lot of language so that they're getting a nice, clear language signal all the time. We want to make sure they're getting a nice, clear language signal. We want to make sure mom is talking to the baby, mom is playing with the baby, and we also want lots of opportunities for babies to interact with a face, with mother's face. So we want mom holding the baby very close, talking to the baby, and establishing eye contact and maintaining eye contact as well as possible. And then getting to a psychologist early for a diagnosis, and then perhaps using applied behavioral analysis in early stages or floor time or other interactive approaches that are designed to normalize the developmental experience of these children so that they can't get off track as easily. Second conclusion is even small changes in the clinical trajectory of many brain disorders can have profound functional consequences. This comes from the article by Gangley and Poo in that, again, that 25th anniversary issue of the journal Neuron that you can all access for free over the next, till November 30th, the next 21 days. Just go into it's Cell Press, go to the journal Neuron at Cell Press, and then click on the 25th anniversary issue, and you can download all these wonderful perspective articles. And the Ganglia article is on neuroplasticity and what drives neuroplasticity and behavioral changes. So the important thing to keep in mind here is we're not talking about parents having to do dramatic things in those early months. And we're not talking about therapies that are years long and hours, 6, 7, 8 hours a day. Small changes, if you have a well-designed neuroscience program, you can get small changes to occur that have a profound functional consequence in a relatively short period of time. So we have the most dramatic program, most dramatically developed neuroscience design programs out there now. The program that was developed at University of Alabama, Birmingham, the Fast Forward programs developed for and tested over 15 years with children on the autism spectrum that can have dramatic effects in a short period of time. And the most effective interventions include a wide variety of computerized approaches that drive neuroplasticity paired with behavioral interventions that address specific functional limitations. So what this means is that we now know what causes autism spectrum disorder. We now know that we have to drive these longitudinal pathways. We have neuroscience-based interventions that get that process going, but we still need the therapist who works with the child, the occupational therapist who works with the behavioral issues surrounding sensory integration problems, the visual therapy that you might be able to get from eye gaze kinds of interventions, the social kinds of interventions that I was explaining to you are being developed with things like virtual reality by Nicole Russo with her visual environmental social information processing approach. And then, of course, the dynamic language and auditory processing issues you can address with forward, paired with good therapy like perspective-taking therapy and good language therapy so that children start using the language that they have and they start interacting appropriately with their language once they've driven those fiber tracts to mature. So that is the end of our discussion, and I can now take some questions. So let me see. I have a few questions here, and before Carrie hops on with some of hers, I am going to get some questions from the participants. So one participant said, new vaccines are new to our environment. How can you assume that current and new vaccine schedules have no impact on ASD? That's a very good question. Actually, there's quite a bit of research now showing that vaccinations in no way affect autism spectrum disorders. Now, those are large group studies, and I should emphasize that. Those are called epidemiological studies. So you look at hundreds of children who've had vaccines, and you look at whether the vaccination schedules that they had in any way distinguish them from children who didn't have vaccinations and the occurrence of autism spectrum. And right now, there is no link. If you're interested in the research on that, it was Wakefield who originally proposed that vaccinations might have an effect on the development of autism spectrum disorders, and Wakefield has since retracted his paper on that subject because he agrees that there were errors in the paper. The concern that doctors have, pediatricians have, is if parents are concerned about vaccines and they either limit vaccines or they schedule one at a time, that the children will not get vaccinated against diseases that kill children, like mumps. And we're seeing a new increase in mumps, you might know, and it can be very destructive, not only to the child's health in general, but to the brain. Measles is very destructive to the human brain. It can cause blindness and deafness.
German measles in a pregnant mother can have a devastating effect on the child's brain. So if children are not immunized against German measles, we could have many more very severe disturbances that are much more catastrophic. Diphtheria causes death. And because there's no evidence that vaccines in any way cause problems, what pediatricians will do if a parent is concerned, if you feel like, well, maybe there's a possibility that because I'm an older parent, or maybe I have a child or an uncle or an aunt or a cousin on the spectrum and I'm a little bit concerned, doctors can break the vaccines up and give them one at a time. But again, I want to emphasize that there's absolutely no research right now linking vaccinations to autism spectrum, whereas there are links to other environmental kinds of influences like petrochemicals, exposure to chemicals released when oil is refined, and also chemicals associated with pesticides. Okay, let's see. Another question raised is vaccinations also impact inflammation due to immune factors? Yes, they do. What's interesting, though, in the new research is that it is the actual virus that the mother has, the infection that the mother has, that triggers the antibody inflammatory response. So vaccinations are a way to prevent that. And again, no research linking vaccinations to autism spectrum disorder, but if it is a concern to anyone out there or any parent, certainly they can talk to their pediatrician about having the vaccinations separated, so they're not getting a full dose of 3 vaccinations at the same time. Another question that came in is, since the self was used with pre and post testing, do you know why age levels were used for comparison, not standard scores, and are standard scores available? I'm pretty sure, yes, standard scores are available. I think the reason that age scores were used is because most people understand age better than standard score. So most parents, most teachers, most lay professionals who don't know a lot about language testing understand age better. So it's a little easier to explain that to parents, but if you have an age score, you do have a standard score, and I think those are available, and I'm sure I can get those from our original research trial. I'm not sure about the Poglich, Marcy Melzer research, but I think we probably could get that too for you. All right, and then let's see, another question. So many pediatricians and even neurologists send parents away before the child is age 2, thinking that autism can't be diagnosed that early. How can they be taught about these breakthroughs? Actually, that's an excellent question. Thank you. And actually, we have, there are early diagnostic guidelines now that were published for before a year, and those have been disseminated through the pediatric journal, the Journal of the American Pediatric Association. So physicians, pediatricians do have access to that information, but in your area, one thing you could do to really help this would be to get those guidelines, and I can get them for you if you want, and disseminate them to pediatricians in your area if you know of pediatricians who aren't yet aware of those. But most of the pediatricians I know around here are looking very carefully and screening children very carefully, starting at that year checkup. Next question came in. Can you please put up the slide with that 25th year reference link that's free until November 30th? I can do that. Let's see, it's going to take me a minute to get there, or I can just tell you how to get there. What I'm going to tell you to do is you're going to go to Internet Explorer, or however you go to the web, and in your Google window, type in Cell Press, C-E-L-L, Press. And then what you're going to do when you get to Cell Press is you're going to see a list of journals, and you're going to pick the journal Neuron, N-E-U-R-O-N. And I want you to click on that journal, And if you do it today or tomorrow, the new neuron issue hasn't come out yet. They come out every 2 weeks. So this 25th anniversary issue will be the issue that you get at Cell Press when you click on the Neuron Journal. Click on that journal, and you can get all of the perspective articles for free. And those are the ones you want, because those are the summary neuroscience articles. So thank you for asking for that. Let's see, the next question is that I'm seeing come up is, in addition to autism, my 15-year-old daughter has a diagnosis of moderate mental retardation. Would fast forward be a benefit for her? Yes, actually, we have research on autism spectrum disorders, and we also have research from the original research trial on developmental delay. And we also have other research that I could get access to you from school districts around the country who have used fast forward programs with different kinds of developmental delay, from Down syndrome to fetal alcohol syndrome to other kinds of causes of developmental delay. But definitely fast forward is very effective for children with developmental delays. All right, next question. I'm scrolling down here. Were the children that experienced great improvements with fast forward also receiving intensive OT, speech therapy, and other therapies? In the Gigi Poglich, Marcy Melzer study, many of those children were receiving speech therapy, because at that time, when that study was done, almost all the fast forward providers were speech and language pathologists. So I would say, without hesitation, that all the children had been getting some kind of speech therapy. Most children on the autism spectrum who are receiving speech therapy are also receiving or have received occupational therapy because of sensory integration issues. So I don't know the answer to that, but I would speculate a lot of the children were receiving occupational therapy as well. And that raises a good issue, and I'm so glad this person asked this, which is, in a retrospective study, you have lots of variables that could contribute to the children's improvements, and you're not controlling for those. So the Poglich and the Melzer retrospective study was not a random controlled trial, although the outcomes were excellent. We do have some more controlled studies that were done in school districts with children on the autism spectrum. I didn't put those in for you, but those are available on the website. One was done at Schaumburg School District, for example, that were more controlled, that did show equally high levels of gains of children on using fast forward when other controls were in effect. So thank you for that question. Next question. Were the children that experienced, I just read that. Question from Seattle. Are there other computerized programs aside from fast forward which could be used in a school setting? I'm especially interested in learning if there are activities which could be used with 2 children who are needing increased verbal social interaction with peers. Thank you. To increase verbal social interaction, especially with young children, and this person said, doesn't say the age, there will be more programs out there. The one that I mentioned to you is a virtual reality program that's being developed at Rush Neurobehavioral Center here in Chicago, and it is called VSIP. And then there is the program, that the eye tracking program for social skills that showed good outcomes with social skills, that was developed at University of Alabama, Birmingham, and that is when you get the PDF, you can see that article is highlighted for you. So you can look up that research and order that computerized program. So one is available. I think it's called, I want to say CSAY, but I can't exactly remember what it's called. And the other one is the new one coming out, VSIP, and then fast forward, of course. Okay, and then what does research about the effectiveness of fast forward training for teenagers with autism spectrum disorders? Well, in fact, actually, we have quite a bit of research with teenagers. Several of the children, many of the children that were studied in the Poglich-Melzer study were actually adolescents. We also have, you can go to the website for scientific learning, www.scientificlearning.com, and click on the results tab, and you can see research on adolescents in general. But I can tell you that in the both in the National Field Trial and in the Poglich-Melzer study, adolescents were included. And I will tell you, in my own experience, having used Fast Forward programs with children on the autism spectrum now for 14 to 15 years, and I was in the original research trial, and I had several children on the autism spectrum, and several children were adolescents, and I've had many adolescents since then, and they do respond very nicely to the Fast Forward programs. And the nice thing with adolescents is there is an adolescent-based program called Literacy and Literacy Advanced, and some more advanced reading programs that actually get into reading or into reasoning, so you can get at some of these higher level skills as well as the low level perceptual skills for adolescents. Okay. What does, that was the one I just read. It keeps going backwards on me. I would love the guidelines, please. Okay, so what I can do is I can send those, I think what she means is the guidelines for pediatricians at the first year of birth, and those were published not so long ago, a couple of years ago, but I can include those as a PDF with the slides. So if you go to webinars at SciLearn.com, I'll have those guidelines for pediatricians for early identification of autism spectrum available as well for you. I find children on the autism spectrum using Fast Forward sometimes become overstimulated with the graphics and animations, then need redirection. Any suggestions? That is a very good question. And yes, I've seen the same thing. Sometimes what I'll do if a child is overstimulated is reduce the protocol to a shorter protocol. So if the child is on the 50-minute protocol and it seems to be overstimulating them, I've had some children start rubbing their hands, or I had one little boy who chewed the collars on his shirt, just showing little signs of overstimulation. You can use a shorter protocol. You can go to the half-an-hour protocol if you want, which is always an option on Fast Forward. You can also, if you think that the stimuli are a little overstimulating for the child, I know a lot of providers will cover up the screen, for example, and just have the child, for example, on SkyGem, just have the child listen to the sounds and tell them what they hear, high, low, or low, high, not actually look at the screen at the same time, or cover up the screen in the beginning when a child's first using Fast Forward, and then gradually introduce the visual stimuli as the child is able to tolerate that. Excellent question. Let's see, is there a way to access Fast Forward and have it covered by insurance, specifically TRICARE? Many providers have used Fast Forward and were able to get it covered as an intervention because they have a medical diagnosis. So there is a medical necessity. You need a doctor to write the script for Fast Forward, usually for most insurance companies. So you need the doctor to write the script, and then you need to be able to document very carefully that what you're working on. And the nice thing about going into My SciLearn is you can get all of the data day by day by day on auditory. You can show auditory perception increases in speech sound discrimination. You can show improvements and document in working memory. You can show documented improvements in language day by day by day and submit those to the insurance company. It would fall under often you would, if you want it covered by Medicare, or sorry, covered by insurance, you would need to sit with the child each day, and it would be covered under speech therapy. So you would sit with the child and actually be intervening while the child is utilizing the program. Let's see what else we have here. At what point do you discharge a child who's been using fast-forward language and makes minimal gains? I've seen a child with very low cognitive skills, severe attention and focus problems, and seizure disorder who's been using the program for over a year. Where do I go from here? Well, the first thing I would recommend is post-testing, because even though children don't make gains on fast-forward per se, they often show gains on tests. So I'll just give you one example of a child I had. When we were first using fast-forward language in the national field trial, we had many children. We only did it for 4 weeks, but then we had many children who could continue on forever. And in those days, we didn't have progress trackers, so we didn't have good ways of documenting how the child was moving toward completion. We had what was called lesson, which gave us a lot of details on the child's improvement, but you just couldn't see how they were moving. And I had several children who showed practically no gains at all for months on what is now SkyGym. Then it was called Circus Sequence. It was a little different graphics. But when I actually tested those children, I saw, even though they hadn't shown gains on lesson and on the data, they showed remarkable gains on testing. So number one, I would, rather than just keeping a child on fast-forward language, let's say for a year, and then trying to decide, should you stop, I would just do some post-testing and see where the child is. Secondly, you can blend. So if a child's really struggling on a couple of exercises, but they've moved along on a couple of other ones, you can blend in exercises from the reading programs or blend in exercises from the language to reading or whatever, literacy advanced, and Heather Myers can really help you with blending so the child doesn't get so frustrated. And then third is intervene. If the child's really struggling, you can take the child offline and do demos with them and really help them and see if that will get to move them along. And again, Heather can help you with that. Now, I have that at 7:31, so I don't know if I should keep going. Carrie, should I, or?
We have a few more questions, so it couldn't hurt to finish up a few more. And then if we have to, people can email us any additional questions that they have.
Okay, so why don't I just answer a few more?
I have a couple too, by the way. I have a couple that have come in just to me.
Should I answer 2 more and then have you answer some?
Sure.
Or yes, some. Okay. So somebody said, I used fast-forward with 2 developmentally impaired 18-year-olds, and the Goldman-Fisco-Woodcock TOAD increased after 4 weeks. Yes, that's very exciting. I've seen many great changes in older adolescents and young adults. And then final question, are you familiar with a computerized therapy? I believe it's called brain state technology, and it has been used with ASD. I am not aware of it, and I don't know of any random controlled studies with it. If you do, please email me. You can get access to me through Carrie at webinars@xylem.com because I'd be happy to look at the data on that. So Carrie, what questions did you get?
I got, let's see here. So the first question I got was, hang on one second. Can typically developing children encounter damage to the white matter fiber tracts at later ages, school age or above, that result in behavioral changes? What might cause these changes, and can environment have an impact on fiber tract development?
Okay, so yes, number one, environment has an effect on fiber tract development. It just does. So fiber tracts, a great study by Stanislaus Dehaene compared the fiber tract maturation and brain maturation of adults in Brazil who were readers versus non-readers. So people who were literate versus non-literate, and showed their brains were very differently organized. So number one is environment affects fiber tract maturation. And that's how the fiber tracts know how to mature. So the way that your brain decides which fiber tracts to invest in metabolically
is based on what demands are in the environment. So yes, processing problems just because of the perceptual abnormality and the fact that it interferes with the way the child perceives either the visual world or the auditory world can interfere with fiber tract development. So you don't need autism spectrum disorders. You can just have auditory processing disorders. And there's neuroscience research on that. Also, other things that impair fiber tract development. So you might have a developing brain that's normal till a child's 12 or 13, and then all of a sudden you see behavioral changes. And one thing we know that can cause that is head injury. So traumatic head injuries, or even just head injuries that have never been diagnosed, that are the result of post-concussion syndrome. So just having a concussion can cause shearing strains, which actually shear the myelin on long fiber tracts and impair processing. And that's well known. And then we also have demyelinating diseases that can affect fiber tracts at a later age, like multiple sclerosis. So yes, fiber tracts are vulnerable at all sorts of ages, and autism spectrum disorder isn't the only kind of disorder that can affect them. Good.
Okay, great. Next question. This had something to do with, you were talking about doctors writing a prescription and this woman is asking, will a doctor write a script for a commercial program?
Well, the doctors will write a prescription often for speech therapy, and then it's for you to determine if you're a speech therapist, or for occupational therapy, if you're an occupational therapist, or if you're an educational therapist. So if you receive a script for that kind of therapy, then it's up to you to determine what the therapy is that you use. And fast forward language especially is a very intensive form of computerized speech therapy, among other things. So all you need is the script from the physician saying that there's a medical necessity for improving perceptual skills or improving language skills. And then it is up to you as a speech pathologist to determine how you intervene and what interventions you use. But I will caution you, if you're going to try to get insurance coverage for fast forward, you've got to be doing speech therapy. You've got to be sitting with the child. You can't have the child be using fast forward at home under the parent's supervision. You need to actually be with the child and actually be sitting there working with the child during the intervention.
Okay, great. The next question is, someone asked if you could talk about that magazine article again, the Neuron.
Sure. Okay. So one more time. I know it's late at night for some of you, and I'm taxing your working memory. So the journal is Neuron, Neuron. It is the October 30th issue of Neuron, which is the 25th anniversary issue. The way that you can access it for free through November 30th is you go to your Google window. That's the only way I know how to do a search. I know you could Yahoo it too, probably. So, but you go to your search engine, you type in Cell, capital C-E-L-L, Press. When you get to Cell Press, there will be several journals, and you click on the journal Neuron. And if you do it today or tomorrow or the next day, Neuron comes out twice a month. And so that October 30th issue is the issue you will get when you click on Neuron, the journal. Click on that issue, and you will see that you have free access to the Perspectives article. You don't get access to the entire journal, but you get access to these 7 or 8 Perspectives articles, which are a history